Hey friends, in this session we are going to read nucleotides, chemistry and metabolism. Okay. Nucleotides are precursors of nucleic acids, DNA and RNA. Nucleotides are also components of important coenzymes like NAD+, FAD and metabolic regulators such as cyclic AMP and cyclic GMP. Composition: 3 components: nitrogenous base, a purine or a pyrimidine, pentose sugar, and phosphate groups. This phosphate group give DNA its negative charge. Due to this phosphate group, DNA is negatively charged. Okay. So this is a structure of what I can say RNA. Bases present in nucleic acids: 2 types of nitrogenous bases, purine and pyrimidines. Purine bases: adenine 6-aminopurine and guanine 2-amino-6-oxopurine, 2-amino-6-oxopurine. So here you can see adenine 6-aminopurine. So numbering is like this: 1, 2. So you have to move like this: 1, 2, 3, 4, 5 and 6, 7, 8 and 9. Okay. So like here in adenine at 6th place amino group is present, so 6-aminopurine, and in guanine 2-amino because 1 and 2. So here you can see this 2-amino, and 6 on the 6th place oxygen is present, so 6-oxopurine. Okay. Guanine is 2-amino-6-oxopurine and adenine is 6-aminopurine. Other minor purine bases are hypoxanthine, xanthine, uric acid and caffeine. 6-oxopurine because when we will name it, like here you can see 1, 2, 3, 4, 5, 6, 7, 8, 9. So on 6th place oxygen is coming, so 6-oxopurine, and 2,6-dioxo. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So on 6th place and on 2nd place oxygen is coming, so 2-dioxopurine. Okay. And here 2,6,8-trioxopurine, 1, 2, 3, 4, 5, 6, 7, 8, 9. So on 2nd position, on 6th position, on 8th position oxygen is coming, so 2,6,8-trioxopurine. Okay. Pyrimidine base: cytosine, uracil and thymine. Just learn it by this mnemonic: cut is for pyrimidine bases. So thymine, cytosine and uracil. Other modified pyrimidine bases are dihydroxyuracil and 5-methylcytosine. Okay. And how we will name these like this: so 1, 2, 3, 4, 5, 6. Okay, no, like not like this. We have to move like this: 1, 2, 3, 4, 5, 6. Okay, in cyclic manner we have to move. Basic names of nucleosides, nucleotides and N-triphosphatase. Nucleosides: adenine plus ribose, adenosine; guanine plus ribose, guanosine; uracil plus ribose, uridine; cytosine plus ribose, cytidine; hypoxanthine plus ribose, inosine; xanthine plus ribose, xanthosine. Deoxyribonucleosides: adenine plus deoxyribose, deoxyadenosine; guanine plus deoxyribose, D-guanosine; cytosine plus deoxyribose, D-cytidine; thymine plus deoxyribose, D-thymidine. And nucleotides are adenosine plus pi, adenosine monophosphate AMP, adenylic acid, guanosine plus pi, guanosine monophosphate GMP, guanylic acid, cytidine plus pi, cytidine monophosphate CAMP, cytidylic acid, uridine plus pi, uridine monophosphate UMP, uridylic acid, inosine plus pi, inosine monophosphate IMP, inosinic acid, deoxynucleotides, D-adenosine plus pi, DAMP, D-adenylic acid, D-guanosine plus pi, DGMP, D-guanylic acid, D-cytidine plus pi, DCMP, D-cytidylic acid, D-thymidine plus pi, DTMP, D-thymidalic acid, nucleoside triphosphate, nucleoside monophosphate, nucleoside diphosphate, nucleoside triphosphate, adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, AMP plus when we will add a pi to it, like add a pi, so just add a pi. So AMP is converted into ADP and ADP is converted into ATP. Guanosine GMP, GDP, GTP, inosine IMP, IDP, ITP, cytidine CAMP, CMP, CDP, CTP, uridine UMP, UDP, UTP, DAMP, DADP, DATP, DGMP, DGDP, DGTP, DCMP, DCDP, DCTP, DTMP, DTDP, DTTP. Nucleosides and nucleotides. Adenine, sugar is ribose and nucleoside is adenosine. Phosphoric acid, 5th position, nucleotide AMP. Same like this, 3rd position, 3rd prime AMP. Deoxyribose, D-adenosine, 5th position, DAMP. 3rd position, 3 prime AMP. Cytosine ribose, cytidine, 5 position CMP, 3rd position 3 prime CMP. Deoxyribose C, cytidine, 5th position DCMP and 3rd position D 3 prime CMP. Now, biosynthesis of purine nucleotides. It can be explained in two different paths, de novo pathways and salvage pathway. Biosynthesis of purine nucleotides. Dust bin pathway. De novo purine synthesis. It involves three main steps: ribose biphosphate to IMP synthesis, IMP to AMP synthesis, IMP to GMP synthesis. Ribose 5-phosphate to IMP synthesis. Amination. Starting material for purine biosynthesis is ribose 5-phosphate, a product of HMP shunt. Ribose 5-phosphate is converted into phosphoribosyl pyrophosphate by pyrophosphokinase. Addition of N9, incorporation of C4, C5 and N7, addition of C8, addition of N3, cyclization, closing of ring, addition of C6, addition of N1, removal of fumaric acid, addition of C2 and cyclization. So here is the steps of PRPP synthase, PRPP glutamyl amidotransferase, formyl transferase, synthetase, carboxylase and synthetase. So one trick is like this: A, B and C. So whenever there is these terms, then what we can learn is A for ATP and B for biotin, and C for carboxylation. Okay, and so this is the fact ABC. Okay, so adenylosuccinase and then formyl transferase and cyclohydrolase. Inosine monophosphate is formed from there of the alpha ribose 5-phosphate. Donor atom glutamine N9, rate limiting Product is PRA, glycine, C4, C5 and N7. ATP required, methylene THFA, C8, FGAR, glutamine, N3. ATP required is FGAM. Ring closure, ATP needed, carbon dioxide, C6. Aspartic acid, N1 and formyl THFA, C2. And for N1, ATP required and fumarate removed. Ring closure. IMP. IMP to AMP. IMP is the central intermediate of both AMP and GMP. IMP to adenylosuccinate by adenyl succinate synthase. Adenyl succinate to AMP. IMP to GMP. IMP to XMP. This is dehydrogenation via enzyme IMP dehydrogenase. XMP is converted to GMP by enzyme GMP synthase. Here amino group donor is glutamate. Ribose 5-phosphate